Hello, everybody. My name is Imke Schmitt. I work at Senckenberg Biodiversity and Climate Research Center and Goethe University in Frankfurt in Germany. Today, I will talk about abrupt genomic breaks in microbiome populations along elevation and associated changes in green algal and bacterial communities. The question we ask in this study is, how does a lichen holobiont respond to the environment? And with a lichen holobiont, I mean a microecosystem consisting of a fungal partner, the Photobions, and the bacterial communities. And I want to know whether this microecosystem, just like other ecosystems, responds to abiotic environmental factors. Specifically, we want to analyze whether changes in this holobiont are gradual or abrupt along elevation, and whether the responses are coordinated among the three groups of organisms. To address these questions, we need a gradient that covers different climate zones. And we found such a gradient in the Sierra de Gredos in central Spain. We used indices based on bioclimatic data to map the bioclimatic zones along this gradient. And we found that this gradient has three major bioclimates: the Mediterranean, the cool temperate, and the alpine. Further, to address our question, we need taxa that have broad elevational distributions and that form large populations. And this is given in Umbilicaria pustulata and Umbilicaria hispanica that grow along this gradient and that gradually replace each other as you go up the mountain. And you see here in the center of the distribution that the two species overlap. So there is a zone of sympatric occurrence. And we sampled these taxa at 9 different elevations that were spaced approximately 200 elevational meters apart. And for each population, we sampled 100 individuals. These 100 individuals were pooled to a so-called population pool and subjected to pool-seq sequencing using Illumina. This creates a metagenomic set of sequences, and this was then further analyzed by mapping it against the reference genome of the fungal partner. So we have genomes for Umbilicaria hispanica and pustulata available, and we use this metagenomic sequence fragment set and map it against the fungal genome. And this set of sequences is then basically a genomic cross-section of the fungal population. We can use this to assess the genetic population differences among all the different populations. We did this and we found that there is a large number of single nucleotide polymorphisms, so mutations, that differentiate the individual populations. In Hispanica, 200,000 SNPs, in Pustulata, 165,000 SNPs, and these SNPs were distributed rather evenly across the genome. We found them in non-coding regions, but we also found them in functional areas of the genome. And what was really striking is that both fungal host species showed a really similar pattern here. So we have two genetic clusters in Hispanica and in Pustulata. And when we map this on the gradient, you see that the clusters constitute a low elevation group and a high elevation group in both taxa. Now we move on to the photobionts. We assessed the diversity of photobionts along this gradient using ITS meta-barcoding. This is data that was already generated in the context of a different study. And we found that this gradient is home to 5 different Trobuxia lineages, 1, 2, 3, 4, 5. And interestingly, these 5 lineages are not randomly distributed along the gradient. For example, you see that OTU3, the middle bluish color, appears only in the 2 bottommost population. OTU number 2, the light blue color, appears only in the central populations. And OTU number 4 and 5, these dark blue colors, occur only in the topmost populations. So there is a kind of niche partitioning going on along this gradient concerning the algal symbionts. Now we look at the bacterial communities. We assessed bacterial diversity using 16S meta-barcoding. And the first thing that we saw was that diversity of bacterial communities decreases with increasing elevation. This is shown using different diversity measurements, and we see it in each fungal host individually. The orangish is pustulata and the blue is hispanica. And we also see it in both hosts combined. So this is a pattern that is well known also from macro communities, that we have decreasing diversity with increasing elevation. Further patterns that we observe in the bacterial communities are these, is this structure here. So here you can see that the bacterial populations form 3 rather distinct groups along the gradient. And the environmental conditions are given down here, where you see that the gradient is spanning cool and wet to hot and dry conditions. And the bacterial communities separate along this same axis. Now, you remember that in the center of the gradient, the 2 host taxa overlap. So this was a good opportunity for us to assess whether the fungal host species also plays a role in structuring the bacterial communities. And yes, when we observed only these, the populations in the overlap zone, and we assess the influence of the fungal host, then we see that there are 2 clear groups, pustulata and hispanica. So we conclude that the environment as well as the fungal host drive the bacterial structure. Now we try and put it all together. In this first part of the graph, you see the genetics of the fungal host, and you remember that there was a strong break here, genomic break here in the fungal partner pustulata and in the fungal partner hispanica. Then we see the bacterial communities, and there is a break within fungal host in pustulata as well as in hispanica. And there is also some differentiation between hosts. And then there are the algal symbionts that show these differential occurrence along the gradient, with some taxa never crossing particular altitudes. So the changes that we do observe here are not happening at exactly the same altitudes. But when we step back a little, we can observe 2 zones that apparently harbor a lot of change. And these zones are located between 1,500 and 800 meters, where we observe a genomic break within hispanica. We observe the upper distribution limit of pustulata, we observe the upper limit of the algal OTU2, and we see a bacterial community turnover. And then there is a second belt further down, 800 to 1,200 meters, where we observe the genomic break within pustulata, the lower distribution limit of hispanica, the upper limit of algal OTU3, and a second bacterial turnover. So apparently there are two zones characterized by 11 to 13 degrees average annual temperature and 7 to 9 degrees average annual temperature, where a lot of changes are accumulating. So these could be two ecological zones that comprise conditions which make it impossible for certain species or lineages to cross. And when we sum up, we can say change along the gradient can be really abrupt. We can see that the lichen holobiont responds as a unit. So all three components that we analyzed here do respond to the gradient. We observe that the changes and limits concentrate in two elevation belts, and we can speculate whether these elevation belts constitute ecological boundaries. What remains to be seen is whether these potential boundaries are universal, that is, whether they occur in other taxa and along other gradients, and whether they might be present in macro communities, such as vascular plant communities, as well as in micro communities, such as lichens. And with this, I would like to show you pictures of the other authors of this study, especially Gregor Rohlshausen, who did all the analysis of this study and produced most of the figures that I showed you today. And I would like to thank you for your attention, and I'm looking forward to taking your questions. Thank you.